Buenos días o buenas tardes. Estamos en la frontera esa del día y la tarde. Quiero darles a todos la bienvenida a esta 54ª edición de la Lección Conmemorativa Jiménez Díaz. Les recuerdo que esta lección conmemorativa la instaura la Fundación Conchita de Jiménez Díaz al año prácticamente del fallecimiento de don Carlos Jiménez Díaz. Crea esta fundación su viuda Conchita, y una de las misiones que tiene es crear esta lección en memoria de don Carlos Jiménez Díaz. La primera, por cierto, la dio nuestro premio Nobel, el primer premio Nobel en medicina español, Severo Ochoa. Sin más, quiero presentar a quien va a dirigir este acto, que es la doctora Raquel Lloti. Nos ha acompañado hace 2 años también en esta lección conmemorativa, en la época solo del online. Ahora hay muchas personas también conectadas online, a las que también saludamos. La doctora Lloti, en los últimos años se ha dedicado a la gestión de la investigación como presidenta del CSIC, ahora como secretaria general de investigación del Ministerio de Ciencia e Innovación, pero es una conocida cardióloga, ¿no? Sobre todo en imagen cardíaca, y le doy la palabra.
Muchísimas gracias. Estimados invitados, científicos, autoridades, es un honor y un verdadero privilegio presidir esta 14ª Lección Conmemorativa Jiménez Díaz. En este día tan especial nos reunimos para rendir homenaje a la destacada figura del doctor Jiménez Díaz y para reflexionar sobre su legado en el campo de la medicina. Fue la persona que cerró un círculo en la medicina, haciendo que en nuestro país pasáramos de una fase en la que la medicina era quizá puramente asistencial e incluso dogmática, como leíamos en el documento que se acompaña a esta lección conmemorativa, a una medicina que incluyó por primera vez, de una manera muy bien estructurada, su dimensión científica y su dimensión también docente. Esa medicina completa en la que muchas de las personas asistentes a esta lección conmemorativa, veo a muchos científicos en la sala, han dedicado su vida. Quiero destacar, en primer lugar, la importancia de la ciencia en el contexto de la pandemia global que hemos enfrentado. La ciencia, todos los que estamos aquí sabemos que desempeña un papel fundamental en la comprensión, la prevención y el tratamiento de las enfermedades. Pero durante los tiempos más difíciles que posiblemente hemos enfrentado a lo largo de nuestras vidas y los tiempos más difíciles que ha enfrentado la sociedad, fue toda la ciudadanía consciente de ese papel de la ciencia para aportar soluciones, caminos de salida a una situación tremendamente complicada como la que vivimos en el caso del COVID-19. Y la pandemia nos recordó la necesidad de confiar en la evidencia científica, en la investigación rigurosa, también en la colaboración internacional para abordar los desafíos de la salud. Los científicos de todo el mundo que han trabajado incansablemente para desarrollar vacunas seguras y efectivas en un tiempo récord, así también como para investigar y comprender los mecanismos del virus y las mejores estrategias de prevención y tratamiento, nos aportaron esos elementos que eran indispensables para poder estar hoy aquí con todos ustedes de forma presencial y no como hace 2 años que tuve el honor también de participar, pero de una manera en la que la distancia aún era patente. Y en momentos como este, en los que se hace evidente el valor de la ciencia, tenemos con nosotros a una invitada muy especial. Quiero reconocer y agradecer y también compartir con todos ustedes la emoción que personalmente me supone el tener la oportunidad de compartir esta mañana con la premiada Katalin Karikó, como saben todos ustedes, una bioquímica investigadora tremendamente brillante que ha dedicado su vida a demostrar cómo la tecnología de ARN mensajero puede constituir una herramienta de enorme valor, desde luego en el desarrollo de vacunas contra la COVID-19, pero incluso más allá. La doctora Karikó, como saben, ha sido fundamental en el desarrollo de las vacunas basadas en ARN mensajero, que han demostrado que son una herramienta de enorme valor en la lucha contra la pandemia COVID-19. Pero además, sus investigaciones han allanado el camino para el desarrollo de otras terapias en otros campos. La presencia de la doctora Karikó en esta lección conmemorativa es un testimonio vivo también del legado de Jiménez Díaz y de su espíritu innovador y de excelencia científica que caracteriza este evento. Su trabajo ejemplar nos inspira a todos a seguir explorando nuevas fronteras en la medicina y buscar soluciones innovadoras para los desafíos de salud que enfrentamos. Y aprovechamos esta oportunidad única para aprender de la doctora Karikó y para compartir conocimientos y experiencias que nos impulsen a seguir avanzando en la ciencia y en la medicina. Una vez más, quiero organizar mi agradecimiento a los organizadores, a la Fundación Conchita Rábago, por organizar una sesión de este extraordinario nivel científico y por brindarnos, desde luego, la oportunidad de compartir esta mañana con la doctora Karikó. Espero que tengamos todos una enriquecedora lección conmemorativa, estoy segura de ello, estoy segura que va a ser además muy inspiradora y que este espacio continúe siendo un espacio de aprendizaje, de inspiración, desde donde honremos el legado de Jiménez Díaz y el reconocimiento del impacto positivo de los líderes científicos en nuestra sociedad. Y sin más, queda inaugurada esta lección conmemorativa Jiménez Díaz. Muchas gracias.
Gracias, doctora Lloti, por presidir este acto e inaugurarlo. Ahora doy la palabra al doctor Gregorio Rábago, presidente de la Fundación Conchita Rábago de Jiménez Díaz.
Gracias. Su apoyo incondicional a la Fundación. Esta es la segunda vez que está con nosotros y le agradezco sinceramente su presencia con nosotros. Al doctor Sastre, su devoción, su amistad durante tantos años y volcarse en organizar un programa que hoy considero que ha sido un verdadero éxito. Enhorabuena. Al doctor Javier Arcos, el director médico de la institución, su apoyo constante a la Fundación. En el fondo somos fundaciones hermanas y yo espero que esto sea el principio de un largo camino. Y luego a Luis Jiménez Díaz, nueva incorporación a nuestro patronato, que ha aportado sangre nueva, ideas nuevas, y le agradezco profundamente esto. Y a todos ustedes por su presencia con nosotros por año tras año. Este año es la 24ª elección conmemorativa y ha sido un placer durante todos estos años. Yo llevo 32 años llevo al frente de la Fundación y es un verdadero placer estar aquí. La lección conmemorativa, como ustedes saben, es un homenaje a una de las personas claves en la medicina española, al profesor o al doctor Carlos Jiménez Díaz, y normalmente con este homenaje intentamos explicar y culminar su vida. La idea es entregar un premio a un científico de renombre nacional o internacional. Y como ha dicho el doctor Sastre, empezó en el año 69 con el profesor Severo Ochoa, quien impartió la primera lección conmemorativa. Me gustaría explicarles cómo nació este galardón y de dónde surgieron las ideas. Don Carlos Jiménez Díaz fallece en 1967 y su mujer Conchita Rábago, el que fue el soporte durante toda su vida y el alma mater de don Carlos, recoge el testigo y, auspiciado por 3 personas, el doctor Severo Ochoa, el doctor Sols y el doctor Grisolía, y por 3 de sus sobrinos, Mariano Jiménez Casado, Pedro Rábago y Gregorio Rábago, mi padre, deciden que los fondos de don Carlos Jiménez Díaz debían destinarse a mantener su nombre y ayudar lo que él consideró durante muchísimos años que era fundamental, la investigación. De esta manera, en mayo del 69, realmente fue en el 68, cuando se crea la Fundación Conchita Rábago, se establecen los estatutos y se le dota de fondos suficientes para continuar desarrollando su labor. El objetivo eran 2. El primero era preservar la memoria de don Carlos y crear el premio Lección Conmemorativa Jiménez Díaz. Este premio homenajeaba a científicos nacionales e internacionales de prestigio reconocido. Hoy ha sido un placer ver cómo los 3 pilares fundamentales de don Carlos, que era aunar la investigación, la docencia y la asistencia, se han centrado en un problema que hemos tenido hace tiempo, como ha sido el COVID. Esta mañana en las charlas que hemos tenido se han visto esos 3 parámetros fundamentales que nos han permitido salir de esta pandemia. Sin la asistencia, sin la investigación y sin lo que hemos aprendido y hemos enseñado, yo creo que no hubiéramos salido de esa lección. Y eso es lo que hace muchos años don Carlos creó. El otro aspecto de la Fundación es fomentar la investigación, la formación biomédica tan importante como se ha puesto esta mañana de relieve, para conseguir avanzar esa investigación básica que es necesaria para conseguir entender qué es lo que pasa y conseguir mecanismos suficientes para salir adelante. También tenemos un programa de ayudas para becas en el extranjero para profesionales médicos que quieren completar y formarse en el extranjero. Cada año tenemos un galardonado que imparte una lección conmemorativa y, como he dicho hoy, ya hace 54 lecciones conmemorativas desde que se constituyó en 1969. Hemos tenido todo tipo de premiados, todo tipo de científicos que han sido personas muy conocidas y simple es el ejemplo de Luc Montagnier, que fue premio en el 88, Príncipe de Asturias en el 2000 y Premio Nobel en el 2008. Yo espero que esto continúe siendo una tradición y que nuestros premiados continúen evolucionando y teniendo los reconocimientos que ellos merecen. Hemos tenido con cada lección conmemorativa un pequeño simposium en el que han participado más de 250 ponentes, jornadas muy interesantes sobre el tema específico, con más de 13.000 profesionales que han ido asistiendo a estos actos. Hoy teníamos aproximadamente unos 200 online y había aquí aproximadamente otras 200 personas. Es decir, estamos teniendo un impacto muy importante, de lo que me siento muy orgulloso. Ya hemos mencionado que el primero que inauguró esta larga lista de premiados fue el profesor Severo Ochoa, al que tuve la suerte de conocer, y detrás de él vinieron otros muchos, hasta 54, con excepción de un año, que fue el famoso año de la pandemia en 2020, que nos vimos obligados, y fue la primera vez que esto ocurría. La razón era clara, a no otorgar el premio, y tuvimos que hacer un pequeño parón de un año. Pero desde que se fundó, ha sido año tras año, hemos ido teniendo gente y premiados. Como saben ustedes, es uno de los premios científicos más prestigiados en nuestro país y, además, gracias al Comité Ejecutivo o al Comité Científico de la Fundación Conchita Rábago, que se reúne periódicamente, elegimos a un candidato que será el galardonado del año siguiente. La Fundación, como les he dicho antes, empezó de una idea de Conchita Rábago, que se la transmitió. Hubo 3 personas que fueron sus 3 sobrinos que mantuvieron y perduraron este legado, que lo transmitieron a la segunda generación, en la que yo me incluyo, y que recientemente hemos tenido una serie de cambios. Marta durante muchos años fue nuestra gerente. El año pasado tuvimos una serie de cambios. Bueno, al final la edad nos pasa factura a todos y esta es la nueva fundación donde está la doctora Rábago, está Luis Jiménez, que se acaba de incorporar como secretario, Pedro Rábago González, que todavía continúa aportándonos información y ayuda, la doctora Ayuso y nuestra nueva gerente Isabel Ferreiro. Con todo, intentamos que este legado se mantenga, que este legado perdure. Esta es nuestra sede y la idea es que hoy, después de 50 años, esta fundación continúe manteniendo vivo el espíritu de don Carlos y consigamos un futuro muy prometedor, tanto manteniendo vivo su espíritu como investigando y dotando de becas a las personas que investigan en nuestro país. Afortunadamente, hoy tenemos una persona excepcional. He tenido la oportunidad de compartir esta mañana una discusión informal con ella y he quedado gratamente sorprendido por su capacidad de comunicación y por su manera de entender la vida. Creo que vamos a disfrutar con su lección y sin más le dejo al doctor Sastre que haga una laudación. Muchas gracias.
Hace justamente un año, en mayo del 2022, el Comité Ejecutivo de la Lección Conmemorativa Jiménez Díaz y por unanimidad pues concedió el galardón para este año a la doctora Katalin Karikó. Así que yo voy a hacer una pequeña presentación de la doctora. Ella nace en Hungría, en Szeged, en 1955. Prácticamente allí es donde realiza todos sus estudios, se licencia en bioquímica y en el año 82 pues, presenta su tesis doctoral. Ella empezó a trabajar sobre los efectos de alguna manera antivirales, precisamente de cosas que se siguen hablando hoy de los interferones. Está unos años trabajando en el Instituto de Investigación húngaro. Eran años difíciles, recuerden ustedes que esos años todavía Hungría pertenecía al telón de acero, había muchos problemas de todo tipo. Ella, después de aplicar, como me ha confesado, a varios laboratorios de investigación, algunos de ellos españoles, pues consigue que le acepten en Filadelfia, en la Universidad de Temple, y se va con toda su familia allí, con periplos importantes, porque les recuerdo que era el telón de acero. Ella confiesa que tuvo que esconder los dólares, porque solo creo que dejaban pasar $100 por persona, ¿no?, en un juguete de su hija para poder ir a Estados Unidos, ¿no? Allí llega a Temple, está un año, sigue sus investigaciones sobre efectos antivirales de interferones y después se traslada a Bethesda durante relativamente poco tiempo y vuelve a Filadelfia ya a trabajar a la Facultad de Medicina de la Universidad de Filadelfia, a trabajar ya más en el campo del RNA. Estamos ya cerca en los años 90 y allí empieza a sufrir sus primeras decepciones y problemas. Recuerden que los años 90, principios 90, era la época que empezaba la terapia génica, realmente, pero la terapia génica fundamentalmente estaba dirigida a manipular el DNA, no el RNA, con lo cual no era la mejor presentación eso de seguir trabajando en el RNA. Son años muy duros, de problemas, faltas de financiación. Incluso en el año 95 pues es degradada incluso en la universidad y su nivel de investigación y de salario por falta de financiación. Ella confiesa, porque una de sus características es, que diré otras, pero es su humildad, que probablemente es que ella no era lo suficientemente lista, inteligente para seguir estas estas investigaciones. Pero bueno, ella sigue allí en la Facultad de Medicina de Filadelfia, y lo que pasa también en la ciencia y en la vida en general es que ocurren casualidades. Y en una máquina fotocopiadora, según nos dicen, conoce allí en los pasillos a una persona, a Drew Weissman, un inmunólogo que se había formado, venía de formarse con Anthony Fauci, ¿no? Que fundamentalmente estaba enfocado a intentar el tratamiento del VIH. Y de alguna manera de esta unión de ideas, pues él pensó también que este mecanismo de usar RNA podía ayudar de alguna manera al tratamiento del VIH. El primer problema grande que tuvieron que solucionar es que el RNA mensajero que ellos inyectaban era inmunógeno, o sea, que producía unos anticuerpos y una reacción en el propio organismo. Ellos lo solucionaron, ¿no?, con una brillante idea, que es cambiar una de las bases, ¿no?, uridina por pseudouridina, y con eso consiguieron que no fuera inmunológico. Estamos ya casi en el año 2005. Ella y Drew Weissman crean una empresa, la RNA-RX, que dirigen precisamente para, de alguna manera, explotar todas las patentes que hicieron y seguir trabajando en este RNA mensajero. Primero, es verdad que ellos siempre pensaron, y ella siempre pensó, en tratar una enfermedad más que inmunizar, ¿no?, con el mRNA. Estamos ya cerca del año 2010 y resulta que dos compañías empiezan a comprar patentes a ellos y a la Universidad de Filadelfia. ¿Y qué dos compañías? Pues les va a sonar: Moderna y BioNTech. Esto empezaron también fundamentalmente pensando en el cáncer más que en vacunas. De hecho, la doctora Karikó se va a Alemania a codirigir BioNTech, una empresa también fundada por unos investigadores turcos en Alemania, en Mainz, cerca de Frankfurt, y allí pasa muchos años trabajando en la posible utilidad de este RNA mensajero en el tratamiento, sobre todo de enfermedades, no de inmunización. Tenían problemas técnicos que con la ayuda del doctor Lynch resuelven, porque producía pocas proteínas, era muy inestable, que era propuso la idea de encapsularlo en liposomas, ¿no?, que actualmente es la solución que realmente se usa. Todo esto, pues, llegó la época de la pandemia COVID, y gracias a que había, como se ha comentado antes en el simposio, esta base de investigación, pues se pudo realizar una serie de vacunas COVID utilizando esta tecnología del RNA mensajero. Yo creo que esta investigación sigue y ellos siguen trabajando en esto, en enfermedades cardiovasculares, cáncer, enfermedades autoinmunes. Pero bueno, por resumir, yo creo que una característica clave en la doctora Karikó es la perseverancia, ¿no?, la perseverancia entre las adversidades, la incomprensión de la comunidad científica en su tiempo, y ya cuando uno la conoce más de cerca es lo que he comentado antes, que es la humildad de una investigadora. Yo no sé si esta característica está en sus cromosomas porque y tiene herencia autosómica dominante, porque resulta que la doctora Karikó, yo creo que es una cosa interesante de saber, tiene una hija que ha sido campeona olímpica de remo 2 veces y 5 veces campeona del mundo de remo con el equipo estadounidense de remo. Entonces, esto me imagino que también requerirá mucha perseverancia. La doctora Karikó ya confiesa que ha sido transparente durante 40 años, que nadie le hacía caso, y que en los últimos 2 años no deja de viajar y de recibir premios altamente merecidos, por supuesto, ¿no? Ha recibido premios en todos los países, en Japón, en Polerglitz, en Estados Unidos, Benjamin Franklin. Aquí en España saben que es de las fronteras del conocimiento del BBVA, también lo ha recibido, y de la princesa de Asturias, junto con Weizmann y Lench. Y bueno, pues ahora es un honor para nosotros que haya aceptado esta condecoración y haya aceptado dar esta lección conmemorativa Jiménez Díaz sobre su tema, que es el tratamiento con mRNA. Doctora Karikó, cuando quiera.
Thank you very much for this nice introduction, and dear fellow scientists, physician colleagues, member of the foundation, the minister, the ambassador of Hungary to Spain, and the Spain ambassador to Hungary. Thank you very much for coming, and all of those who are online. And today I will talk about developing mRNA for therapy. And as was mentioned, you know, I have some financial interest here, but otherwise, what is important to talk is that for years, we understand that discoveries coming from academia, universities, research institute, and when something seems to be useful, then small companies are formed and testing out the new technology, what it would be useful, and if it is advancing well, you know, maybe larger companies help in if it is a clinical trial, and then we thought that the end is a FDA or EMA approved product, and that is how society benefits from it. But we learned society has to accept that new product, and whether it is a GMO, whether it is new technology, whether it is the mRNA therapy and vaccines, we learned that. So today, the presentation I want to dedicate and ask all of you who are professional that we have to help the public to understand. Education is important, fighting misinformation, and you know, many people ask me, you know, which scientist? One scientist says the opposite, what is the other, and which science I should believe. What is important for the public to understand that the scientific process, because it will correct, and we will learn eventually what is the truth. That's what they have to believe, and we have to talk to the reporters and help them to communicate. And I like to talk to other fellow scientists because I learn from them. But we have to learn also and practice to talk to reporters and explain simple way what we are doing, because otherwise, you know, the public will not accept our discoveries. So today I will help you to understand that the success of RNA vaccine was not an overnight success, but it was many years. Actually, it was 60 years from the discovery of mRNA to the first time when it became accepted in a COVID-19 vaccine, the mRNA became approved. And very similarly to other discoveries, whether it is Cas9 or others, it is always coming the discoveries, and then the scientists try to understand structurally and functionally what does this mean, and then they try to see whether there is some application, testing out, and that also takes decades, or here in this case, 2 decades, and then when they come up with an application, they try to see that whether it could be used for human use, some kind of, and then that's what happened. The last 20 years, actually, mRNA was in a human trial. So as I mentioned, mRNA was discovered in 1961, and there were 2 papers. Interestingly, both describes that this is an unstable intermediate. And I am telling you that there are many, many scientists participated, which eventually led to this product. And here I just show several references here, but even I can say that already the 50s, scientists already, 56, when first time they discovered that messenger RNA, it was a form of phenol-extracted tobacco mosaic virus, they realized that the RNA carries all of this information. And so many scientists participated, discovering that what is the central dogma of life, that we have DNA, which is stable in the chromosome, and in eukaryotic cells in the nuclei, and then it transcribes mRNA and goes to the protein translation place, to the ribosome, and we get the protein. Of course, scientists couldn't make mRNA. They could make RNA. I can mention that, Ochoa was who discovered the polynucleotide phosphorylase, and they could make the homopolymers. And actually, 1961 is a famous year because this was the first time using these homopolymers, we could understand the first code, which was the poly-U was coding for polyphenylalanine. So we learned that thanks to Ochoa discovery. But that was homopolymer, and it was not RNA described from mRNA, so that scientists at the beginning couldn't make mRNA. They could just isolate. So that's what they used. And they selected reticulocytes. This is the precursor of red blood cells because it expressed a lot of beta globin and globin protein. And they could isolate the RNA because so much RNA was present. And then they translated in reticulocyte lysates. They injected to frog oocyte, and they could see that these proteins were made in those lysates or the cells. And very importantly, 1978, there were 2 publications where this isolated RNA was put in the liposome and delivered primary cells, mammalian cells. And somehow this lipid helped to fuse, and then what happened, the protein was detectable in lymphocytes, which normally wouldn't make this globin. I am showing here immunoprecipitation. This was in a Nature paper, 1 author, 1 figures. That was a good time at that time to publish in Nature, so you didn't need so much experiment, but it was very important. So some scientists characterizing the RNA, and other scientists looked at what kind of structural elements are there. So in the 70s, they already discovered the RNA has a poly-A tail. And using this poly-A tail, they invented purification procedure for oligo-DT. And helping with this purification procedure, other scientists focus on and look at the 5'. You see, I am used to work on Apple anyway. So they looked at and they found that there are cap structural elements there. And in 1975, there were like 5 papers on Nature just about the 5' end of the RNA. So it was very important. They already discovered if that cap structure is not there, that the RNA is not translatable. So it was also important that by end of 75, it was Bernard Moss' colleague discovered that there are enzymes, viral enzymes, which can functionally put this cap to the mRNA. And if you receive the Moderna vaccine, they use this enzyme to cap the RNA. If you get the BioNTech Pfizer, that it was transcriptionally incorporated with a cap analog and generated this way. But this was 1975, and in this time, you know, I just entered to the university, and I was eager to do something. And the only place was available is to work in the lipid lab, which seems very boring, yeah? Because I wanted to genetics or something more excited, but it was an exciting time there also because that was when, you know, the cell membrane model of, you know, this sandwich model I learned in school. Actually, we learned just, actually, there is a fluid mosaic model that, you know, the proteins is not just like in covering the lipid, but, you know, crossing the membrane. And I started to work. I went to this biological research center as an undergraduate, and eventually I had my studies these desaturase enzyme. But my supervisor said that, you know, I can spend the rest of my life to work in the bench, and he said that just go to in the summer in this Fisher Institute where farm fish were kept, and I had to collect lipids, and we had to determine which kind of, how saturated or unsaturated lipids are there. I don't say too much about it, not eating too much farm fish after that, but you know, we learned a lot here, and I'm telling all of these things that if you are an undergraduate student, just start with something and made your hands wet, and maybe it is lipid lab or whatever. You just do your best, because what happened in the fall when we went back to the studies and I had to go to school and sometimes I went to the lab, these two scientists approached the lipid team that they want to make liposome to deliver DNA into the cell. And we needed phospholipids. And we were behind the Iron Curtain and we couldn't order it, although it's a couple of Deutschmark you could buy. And we had to go back to an original protocol described 1942 how to make this from cow brain. And we spent a whole week, you know, extracting the cow brain, and it was, you know, handy that my father was butcher, so when seeing a cow brain, I was not run away. And we isolated these phospholipids and generated liposomes, delivered DNA inside the cell, and here is shown that it was labeled, the DNA, and show that it gets inside the cell. And so, again, if I wouldn't do this experiment as an undergraduate, you know, I wouldn't be probably here. But it influenced my thinking later when I worked with other fields that, you know, we did this experiment. But I didn't select it to go to the lipid lab, and I didn't select it to go to the RNA lab to make my PhD. Everything was by chance. So I did not make this big connection that now, after lipid, now I have to work with RNA because that's the future. I don't know at that time, so I just want to make my PhD. Happened that my supervisor, Jenő Thomas, actually made cap analogs and sent to New Jersey to Aaron Shotkin because he needed reference material. So when I started here in 78, I already knew that RNA has a cap. And, but he said that my role will be to make this 2' 5' oligoadenylate, because the prior year, 77, Ian Kerr in London just discovered that this molecule is probably responsible for the interferon-induced antiviral effect. And Jenő came from the industry to this biological research center, and he said, we need an antiviral compound. You know, it was 78. Yes, we need it very much. And so my role was to synthesize chemically enzymatically. And here I show my colleague, János Ludwig, who was a fellow graduate student. Now I am telling to the graduate student, be nice to the fellow graduate student. Even today I ask help. He's an organic chemist, and I need his advice, yes? So we started to make this molecule, and to know this oligoadenylate activates an enzyme called RNase L, which is a latent enzyme. And the idea was that when this small molecule, this trimer, will activate, it will degrade the RNA, specifically the viral RNA, and this is how it exerts antiviral effect. Normally, we make this oligoadenylate synthase, and again, I am telling that this was when I did my PhD thesis, and later I worked with other things. But I kept following these enzymes in the field. And for example, oligoadenylate synthase RNase L importance we learned for the COVID, that those people who oligoadenylate synthase enzyme, which generate this molecule, is somehow compromised, they are seriously ill. And we learned also recently that the children who get in ICU, actually they also had this enzyme is compromised and causing this severe effect. So we learned all of these things about this molecule, but at that time we didn't know any of these things. So as was mentioned, we ran out of funding, and I had to go to overseas. I applied a position here in Madrid, Luis Carrasco Laboratories, but I couldn't get support, financial support. So I went all the way to Philadelphia. Professor Suhadolnyik wrote a basic book on modified nucleosides, and I already, my PhD thesis was, I used cordycepin because it was easy to make 2' 5' oligoadenylates, because, it is like a deoxyadenosine analog. And I worked in the laboratory there, and we did different kind of analogs, but the problem was delivering to the cell. So we are in the 1980s, and the major problem, as we heard today, was HIV. And eventually, we couldn't make this 2' 5' link molecule very effective, so we moved to use double-stranded RNA, which was a mismatched double-stranded RNA to treat HIV patient. And this was 1986. You know that interferon, we just heard good introduction today, was discovered in 1957. 63, we already know double-stranded RNA can induce interferon. We are in the 80s, we still didn't know how. Up until 2001, when TOL3 receptor was discovered, we didn't know how it does the system, but we use this to treat patients. Unfortunately, they made RNase L inhibitor. The virus was resistant, and it didn't help the patient. But I already, as you can hear, I work with long RNA. We synthesize these double-stranded, mismatched double-stranded RNA. I work with short RNA. We labeled it, and we did a lot of things. And but I did not work at that point with mRNA. The field on this area is by 84, had a major advancement, and that was that we finally could synthesize RNA by our design. And that accelerated progress. How it happened? Scientists at Harvard, here I listing Green, Maniatis, Malton, and Paul Krieg, but even prior to 84, 82, we already could make mRNA using bacterial RNA polymerase. What they used here is phage polymerase. That was very simple, very high fidelity, and from the piece of DNA, we could make mRNA. Again, emphasize the advancement of science. By 84, the recombinant revolution happened. It happened 73 when people first time could clone one gene to another place, another bacteria. So all of this advancement is there. And when we have a piece of DNA, we put the basic 4 nucleotides, we put a phage RNA polymerase, and the transcription 1 hour later, we could have the RNA, and then we can put on the cell, and 10 minutes later we get the protein, which is encoded. But here what they did, they also get a capping enzyme from Bernard Moss, and they generated human beta interferon mRNA, and this had the cap structure, had the poly tail, and as a result, they injected to the frog oocyte. If you are old enough like me, then you would know that in the genetic department, usually there was a terrarium and big frogs there because they had large eggs and it was easy to manipulate. So that at the beginning, there were not tissue culture. Even the culture was like primary chicken fibroblasts, what we used. So here they used this frog oocyte, injected, and then culturing overnight and taking the medium, they could see that that medium had antiviral effect. So the first time somebody could make an RNA and it translated in a cell and that produced a functional protein, human protein. I might mention here again, I was starting working when I went to University of Pennsylvania in '89 to work with RNA, messenger RNA, and actually I sent a letter to Paul Krieg whether he could send me this plasmid because it has the promoter and other things. And he put in an envelope and put this plasmid in and said, I have even a better one, and just send it. So there were no material transfer agreement. Every scientist wanted to have the other scientists, fellow scientists, and this was so beautiful. Anyway, so scientists get excited that they can make RNA by design. So you could see that in the 90s, they started to publishing more and more using design messenger RNA. And one of the paper which kept me excited, this 92, this vasopressin mRNA, which was Floyd Bloom, was the editor of Science and Script Clinic, so it's high quality research, and they could treat the animals. And that was I wanted to do always, to treat animals, not eventually, of course, human, but using the mRNA coding for therapeutic protein, and that was my goal. And 93, 94, you can see first time messenger RNA was used for infectious vaccines, for influenza. And 95 was the first paper when messenger RNA was used for cancer vaccine. And 96, Eli Gilboa from Duke University, they spin out a company also, and they made a company, and they used the messenger RNA ex vivo treating dendritic cells. It went all the way to became Argos. First it was Matrix, Argos, and they had phase 3 trial, but unfortunately was not successful. But all of these other scientists and papers, I looked at, follow them, and it was not easy in the 90s. There were, not easy way to follow different scientists, but they did not publish more because there was a problem. It seems that with RNA, there is nothing, just problem. What was the problem? That very small amount, short amount of time, very small amount of protein was made, and it was useless. So it was difficult to work, and of course later we also discovered it was immunogenic, but this instability, that it degraded quickly, and so we needed something to improve. And that's what happened on this field, actually. If you follow, is gradually improving formulation and the mRNA. So it was, mRNA was purified better, kept longer poly A tail, and a different kind of particle which was protecting, because RNA is present everywhere. So now we are here in, I'm starting to work with 89 with mRNA, and I went to cardiology department as a clinical department, so as a PhD you cannot be tenured position, but Elliot Barnett and a colleague who was just 8 days younger than me, so I convinced him that mRNA is the future, and he supported my salary, and so he was interested. he did, they did catheterization, bypass surgeries and whatnot, and, he was interested in urokinase receptor. So I cloned it and made a urokinase receptor mRNA. And what was interesting, that time we had this lipofectin, this positively charged lipid, to complex the RNA, delivered to the cell, and voila, the cell knew all of this post-translation modification, glycosylation. This molecule, to be functional, had to be GPR linked, and it processed the end, and it was functional. So that was so amazing that, you just deliver an RNA, and the cell knows what to do. And so we imagine at that point that maybe we can deliver to a blood vessel this mRNA, and let's say when they do a bypass surgeries, this patency of this RNA can be much better, no coagulation or something would happen. So we were thinking every time about application, that what it would be good for. And we were thinking about, because in vitro we could do very well delivering to cell, we were thinking that, maybe we can use it for delivering RNA to the bone marrow, maybe telomerase, maybe older people can give bone marrow because of the extent of life. So it was a constant thinking like that. And so Elliot supported me for 7 years. I couldn't get grant, but when I met in Elliot's lab David Langer, a medical student. And again, I am telling you, those who are working in the lab and have a medical student, they are usually obnoxious. They know everything, kind of, but it was David like that. But he finally wanted to learn from me, and we became good friends. So when he learned that, I'm losing my job, he convinced the chairman of neurosurgery that neurosurgery needs a molecular biologist. And so this was the laboratory there. I worked 17 years. I did all of the experiment there. Because I couldn't get grant, I had no postdoc or helper. I did all of the experiment with my hands. I isolated plasmid, designed the experiment, seed the cell, transfected the cell, and everything. So here, with David, we wanted to treat patient who had stroke. So one idea was he was coming from the patient and said that, subarachnoid hemorrhage, the patient has this vasoconstriction of major blood vessel. And he said he could reach that area. And we figured out that maybe if we deliver mRNA coding for nitric oxide synthase, which generate nitric oxide, which has just millisecond half-life, maybe locally we can have vasodilatation. And so I made the RNA. We tried to find a different corner of US, some laboratories or who had, set up for this kind of analysis and travel here and there, and did a lot of work. And so I was working there, in this lab. Usually I never had a lab coat up until I recently, got famous and they made one. So I did a lot of work here, and actually everything what with David Langer we did, was focusing on treating patient for stroke. Like we try to catabolize different products with DNAs, RNAs, and APRase, nucleotidase, so that when the dead cells are releasing these products, all is inflammatory, and we try to eliminate that. And so it was like we could do an innovation that he knows a field, and I know another field, and we talk each other, and we respect each other, then we can develop a new product. I wanted always to overexpress a protein which is already present. iNOS is normally present, that protein. So I never thought about immune activation up until, as was described, I met Drew Weissman, who was interested to develop a therapeutic or prophylactic vaccine for HIV. And so, I brag about my that I can make RNA, and he was interested to test out, and we started to work together. Again, he was an immunologist, I was an RNA scientist. We talk each other, educate each other, and develop something. So we did together this first study, and he was very happy that mRNA, it delivers it is adjuvant, it activated the immune system. Of course, at that time, we didn't know that it's not good immune activation for good vaccine, and also it carried and coded for the antigen. And so when we looked at the data, you know, he was very happy, but seeing all of this inflammatory molecule which induced by the RNA, I mean, I was very upset because I want to develop a product for the stroke patient. And that's the last thing I want, to have more inflammation to a patient who already had a stroke. And so we set out to understand that why the RNA would activate the immune system. In all of our cells, we have RNA, and it's not activating. Is it that because we added this RNA to these human dendritic cells from outside, and that's maybe all of the RNA is immune activator? So to find out, we did the following experiment. We isolated different kind of RNA out of the mammalian cells. So we had nuclear, cytoplasmic, mitochondrial, and other RNA, and we tested out in these dendritic cells. And what was interesting that, you know, they were not as inflammatory. You know, purple is the synthetic RNA I made, and I thought that I'm making the same what is inside the cell, but some was less inflammatory, and the tRNA was not causing any TNF-alpha induction. And that gave the idea that, you know, here is the tRNA, has all of this modification, nucleoside modification, and I'm just simplifying those things because the RNA is shorter, the ends is different, so a lot of things always comes to mind. I'm just telling you one of the things which led to the discovery that maybe modification is what is causing that it is not inflammatory. Maybe we have to modify the RNA. Yeah, but how to do it? You know, I'm just showing you that at that time, 100 different modification was known, and we didn't know which one is important, all of them or one of them, and how to introduce these. These are enzymes introducing, because in our RNA, all RNA is made from the 4 basic nucleotides, and post-transcriptionally is different modification introduced, or during the transcription in the nucleolus, for example, in the case of ribosomal RNA. And you just couldn't call up Promega to order an enzyme. And the only known thing, we are in about like 2002 right now, the only 2 modification was known. My fellow, Tamás Kis, we lived in the same dormitories. He was the same student in biology, and we even worked in the same place. He discovered that how 2' methylation pseudouridylation occurring. Again, I'm telling to the students to follow what his fellow students are doing, because if I don't know he discovered these things, I couldn't call him up and for help. And of course, you know, I told him that, Tamás, just can you send me some stuff? I have to modify the RNA, but you know, these require special structural element there, which was not present in my RNA. So I have to ask János Ludwig, the other fellow student, that maybe I have to make the RNA different. Maybe I have to order modified nucleotides and try to see that whether the enzyme incorporates, not how nature is doing. For that one was one important thing I have to remember. You know, today we heard about nucleotides analog, ribavirin, and others. It is very important that in 1993, there was a human trial for hepatitis, and 15 volunteers was there, 5 died in the NIH trial when they tested out fluoridin. And this was a fluorinated, iodinated analog, and it took 10 years to discover how could it be that this molecule was fine, even for monkeys, and the rats get 1,000 times higher dose, and nobody gets sick. Why these people died? And it turned out only the human has a nucleoside transporter, which is equilibrative, and in the middle of the coding sequence has a mitochondrial localization signal, and everything was shoveled into the mitochondria. Those people died from acidosis. And when they looked at their, even prior to that, they already treated patients who had HIV, also with this drug, and when they looked at the data, those patients died from, not from the AIDS, but from the drug. So at that point, I told János, I don't care about any unnatural modified nucleotide. I want just naturally occurring ones. So at the end, he identified me these 10 nucleoside triphosphate, which was commercially available, and tried to see that whether T7 polymerase will incorporate to the RNA. So this is how I try to make the RNA. And what I found is that 5 of them not incorporated, and 5 did. And so I could generate this RNA, which contains these nucleoside modification. And of course, the big question was that whether it made those RNA non-immunogenic. And when it tested out in these primary human dendritic cells isolated from the human blood, this was Drew Weissman did. I have to say that with Drew Weissman, we worked shoulder to shoulder because he couldn't get support money much. And so he did the experiment, I did the experiment, and then we lean over and try to discuss what to do next. So he tested out and with isolated cells, and we found that some RNA was, just as immunogenic, even after modification, but some seemed that was not inducing any TNF-alpha. And we made a closer look. We found every time when the uridine was modified, the RNA was not immunogenic. No TNF-alpha was induced. And so it was that point, we were in 2003 or 4 when we discovered that uridine is responsible for activation, and we also identified the receptor. The paper came out showing that TOR7 can be activated by poly-U. So we learned that, and of course we also knew that if we have certain modification, then the RNA is not inflammatory. Of course, Drew wanted to develop a vaccine. You need RNA to be translatable. I want to develop some RNA coding for therapeutic protein. Translation was critical, so we tested out, and for 2-thiouridine containing RNA was not translatable, but most importantly, pseudouridine containing RNA, we get 10 times more protein. So I spent 2 years trying to understand why. But the importance was that we can have an RNA which translates very well and which is not inflammatory. So we could proceed. And in the United States, usually proceed, you have to file a patent even before your paper comes out. You establish a company and apply for a business grant. So because I was demoted at the university, so I thought that I became a CEO of this RNARX that, we can go out, but we did not expect it that we will not get our own patent for our own company, but that's what happened. But here we proposed, this was a company established for modified nucleoside containing RNA, and we proposed that we will treat anemic animals, and we will have high hematocrit, so on, if we use erythropoietin coding mRNA. So if you follow the bike race, you know that EPO is increasing the red blood cells and the hematocrit. So we did this experiment in animals. At that time, we already commercially available new complexing agent was transit, and in vivo it worked very well in animals. We delivered pseudouridine containing EPO mRNA and very small amount. If you work with nucleic acid, 0.1 microgram is a very, very small amount. And that was sufficient that even 4 days later we could measure EPO protein circulating in an animal. And when you inject just the protein, half-life is 2 hours, so that degrades very quickly. And it seems that if modification was not there in the blue line, then the RNA also translated for a while, but it was not detectable. And importantly, the right panel shows that these RNA were translated to a functional protein because the hematocrit increased. And if we injected weekly very small dose, then the hematocrit could be maintained high in the animals. And most importantly, the RNA was not inflammatory, did not induce interferon. And this is again coming up later importance because the interferon is actually interfering with the virus as we heard today, but it also interferes with antibody production. So this is why the RNA, conventional RNA, is not a good vaccine because antibody generation is inhibited by that. So anyway, again, I might say here, several times I was terminated in my position. I wouldn't be here if it wouldn't happen. So again, in 2013, after all of these studies were done, I had to leave the university and ended up at the BioNTech, which was a very small company without any website. And we were not in this fancy building here, but we were on the campus. And today is 5,000 employees there 10 years later. But at that time, we were very small, and I worked together with Ugo Zain and Özlem Türeci, who established this company. And we did a lot of further improvement of the RNA and scaling up. So many things what I did at Penn, had to be, because, I precipitated the RNA. It is not a scalable procedure and had to be adjusted. So eventually, we did codon optimization, optimizing different elements of the RNA, purifying, modifying, and we could reach now that it's not the small amount of protein, not a short period of time, but high amount of protein for significant time was secreted. So it was kind of, the RNA was ready for prime time. And so why it is important you have RNA. You have to know that most of this, protein, first time was, as a therapeutic protein, was introduced 100 years ago. The insulin was the first. It was purified. Then 1982, again, the insulin was the first recombinant protein which was introduced. And so on. At present, more than 100 products are there, which is most of them is recombinant protein. And they are very effective, many different fields, but it is very expensive, the purification and other things, and antibodies and what is used. So why would be good for RNA? Because your body can make the protein. You don't have to figure out how to purify. The RNA is always from 4 nucleotides, and you can mix different RNA coding for different proteins. There is not a problem, no aggregation, no problem. So, and very quickly can be worked, even if a final product is protein, screening on RNA is much better, much faster, and cheap. And so that's why it is important. First of all, everything which is recombinant protein can be replaced with mRNA, and also when you need an intracellular protein, you can deliver the RNA. In that case, you have to find that specific cell which needs that protein which is missing. So it's a bigger challenge because you have to target. So what I did there at the BioNTech, because I always want to develop RNA for therapeutic, so that we did focus on mRNA coding for antibodies. Here I'm showing a publication we did where bispecific antibodies we generated an mRNA, and we could eliminate already established large tumors in animals. I show there in yellow that it is already in human trials. So this human trial is started with these bispecific antibodies. Another antibody which was not bispecific, conventional, and not targeting Claudin 6, but Claudin 18.2 is a cancer-specific product, is also in a human trial and advanced there. Another project which I also participated, we did together with Sanofi. In that case, the mRNA, again, is not if it is vaccine, it codes for antigen. Cancer antigen, viral antigen, then it's a vaccine. Here in that case, the RNA codes for cytokines. And these cytokines listed there, the IL-12, interferon, alpha, GM-CSF, and IL-15. So these mRNAs were injected to the tumor, which was close to the surface, in that case, melanoma. And then you convert the cold tumor to hot tumor. The immune cells are rushed there, and then when they circulate, they can recognize remotely located metastatic places. And that's in animal study, eliminating, from the lung the melanoma. And this is also in human trial, and this is for head and neck cancer and melanoma. While I was working here with my colleagues at BioNTech, my colleague together we work still with Penn, and my colleague Norbert Pad in Drew Weissman's lab developed a vaccine for Zika. You remember that in 2017, the major problem we heard today also was Zika. And here, this messenger RNA was coding for this envelope product, and it was nucleoside modified. It was 1-methyl-pseudouridine. It was formulated by Acuitas, the same it was in Pfizer-BioNTech vaccine has. And what was interesting, when monkeys were vaccinated with this mRNA, very small amount, the same amount that good for mice was good for monkeys, and fully protected the animals when it were challenged with Zika virus. This was a big change compared what was prior to that, when people tried to make a genetic vaccine with DNA, because then unproportionally had to have higher and higher dose with the DNA. So eventually the DNA vaccine trial is milligrams were injected. But here seems that same amount is good for mice and 5 kilo monkeys. So that was a big change. And so as a result, all of this development was that we already aware of that what happened in 2020. Zheng Yongzheng, who professor sequenced in Wuhan that virus, and he told me that in January 5 he put into the gene bank. But nobody was watching that, so he asked, at homes, and put out Twitter that the sequence is there. He told me that's how it happened, if you want to know the truth. And so everybody referencing the January 12, when it became available, the information that what is happening in China and what is the sequence. I have to say, in March already, the first patient was injected, the first volunteer, sorry. Because of course these are subjects, they were not sick. And so it quickly happened, everything. In BioNTech the same, I have to emphasize that 2018 BioNTech signed an agreement with Pfizer to develop an influenza vaccine, mRNA based. So we already knew each other, we already worked on this project, and we were ready end of 2019. So to do the influenza. Of course, right now it is in phase 3 because it was put in the back burner because the COVID happened. So you already watched the television, everybody watched the television, what's happening, so you know that how quickly all of these trials went on, and end of the year, 2020 already was vaccinated some people, and 21 is more. What many people don't know, that prior to this COVID, there already mRNA was already used for vaccination in human trial. I mentioned you that 1993 was in animals, first tested, but in human trial, CureVac used for rabies prophylactic vaccine. And although at the beginning they used protamine and they went up to 650 microgram per intramuscular, but when they did a lipid nanoparticle formulation, they used conventional RNA, then even very small dose was kind of inflammatory. But Moderna did avian flu and Zika, human trial prior to 2020. And many people did not know. Today also I heard that, nothing was tested before. Yes, there were human trials, and lots of things were learned from it. Actually, the avian flu trial, Moderna ran in Germany, because in the US, authorities were not ready yet. And because in Germany there was CureVac, was established 2000, and there was BioNTech, so the authorities were more educated how to handle and what to do. So they run human trial there. Of course, this was 200 participants and not 200 million. And so only those people who were in the RNA field were aware of these trials, and not the public. And of course, now that we are 2023, and what's happening? There, you can hear daily that a new human trial is started with mRNA. More than 150 human trial is running right now using in vitro transcribed mRNA. And these trials, most of them is focusing still, for vaccines against viruses. We heard about RSV. Moderna has in phase 3 for the elderly. They have other RSV for the younger generation. The flu, both Moderna and BioNTech, Pfizer has phase 3, the trial. So these were be approved. HIV, Moderna has 2 HIV trials. One is the human project. One was by NIH, so they designed different kind of trial, and Moderna is running. There is Zika virus, herpes simplex virus, as it was run by BioNTech together with University of Pennsylvania. We learned recently that maybe multiple sclerosis caused by ABV. Moderna has 2 ABV trials. Human trial is ongoing. Recently, they have CMV already advanced phase. They recently announced Nipah, that they will do Nipah vaccine. And for viruses, we heard again today that for shingles, the very effective protein vaccine is available. Shingrix, GSK is making. But it is expensive. I mean, it is 600 or 800 euro. And as I mentioned, the RNA vaccines are very cheap. So actually, both Moderna and Pfizer BioNTech announced that they will make an mRNA-based vaccine against shingles. And in addition to viruses, and there are also bacterial vaccines. And of course, TB is an intracellular pathogen, so that it is kind of, you have to target like it would be a cancer. You have to recognize the cells. And of course, the malaria, which is a parasite. And of course, as the pathogen genomes get bigger and bigger, what to target is more challenging, and to figure it out. We heard today also, the cancer vaccine. All of these companies, CureVac, BioNTech, was based on that they will develop cancer vaccines, and that did a lot of advancement, a lot of learning. And, it takes time. There are different kind of vaccines. There are individualized. We heard recently when it was in the news about, for pancreatic cancer. In those cases, the cancer cell or the tumor could be removed. There were no metastases, sequencing, and based on the sequencing, identifying neoepitopes, where its mutation caused amino acid chain, and then 20 such epitope was included in the vaccine in the BioNTech, and trial where Moderna, I think it used 34 epitopes when they used the individualized cancer vaccine, and they did for melanoma. In any case, it was that the tumor was removed and there were no metastases. And then, based on the sequence information in 6, 8, 9 weeks started the vaccination, and what they could find that they could delay very significantly return of the tumor. So for cancer, of course, I mentioned already the antibodies, the bispecific, conventional antibodies, and cytokine encoding mRNA, and there are also a combination of things like CAR T cells together with vaccination is present. For acute diseases, I mean, for VEGF-A, AstraZeneca and Moderna was already 2018 already injecting patient with nucleoside-modified RNA. They used for patient who had diabetes and they have necrotic wound and helped wound healing because VEGF-A codes for a protein generate new blood vessels. That was the same in elective bypass surgeries. They injected naked nucleoside-modified RNA to VEGF-A coding to the heart. And there are more genetic diseases treated. First of all, which was most successful when they used Cas9 mRNA. Intellia used for transthyretin amyloidosis. Those patients who get this disease, their liver cells are making a toxic protein, and they interrupted this successfully. 3 patients, only 1 treatment was sufficient, and for 2 years later, they're still not making this toxic protein. And this is also a very deadly disease. And more protein replacement therapy are in the making. Mostly these are by Moderna. So what is the future and what is my role here? First of all, I want to inspire the new generation of scientists. Here we are there picked up with Drew Weissman doing some experiment there, and so these are books for children on elementary school, and I also wrote a book about my life in science, which will come out in the fall, and I like to thank two colleagues, mostly at BioNTech, because in the university setting I did not have helpers, postdocs, or students, and of course I also thankful my family, my husband, who never complained if the weekends, I was not cooking that much and more like running to the lab with new ideas. And, my daughter, who, is a 2 times Olympic champion, and at University of Pennsylvania, she graduated also there, but, usually they introduce me to people that, did you know Kati's daughter is a 2 times Olympic champion? I was the famous mom. And nobody said that, did you know Kati is working with mRNA? That was not the news. And when I went to all of these races, that I was introduced that Susan mom. Now that my daughter is coming with me now, she's Kati's daughter. Yeah, thank you for your attention.
Bueno, la doctora Yotti y el doctor Rábago hacen entrega de la medalla de la lección conmemorativa, así como del diploma.